Hi, my name is Rachael Jayne. I'm a PhD candidate working with Professor Alice White at Boston University, and I'm working on Thrust Area 2 of the CellNet ERC program, which focuses on nanomechanics. Our lab uses a type of 3D printing called direct laser writing to make polymer or plastic microstructures. So today I'm going to talk about how direct laser writing works, the challenges that we typically encounter when doing this kind of research, and how it's applicable to CellNet. So a lot of people have at least heard of 3D printing. It's a very quick way to make almost anything that you can think of. You can design a part using a computer-aided design program, a CAD program. You can import it into the printer, and then you can have the design printed a short time later, and you have an actual part ready to go. And so it makes a lot of sense for prototyping custom devices or parts. And there's two main types of 3D printing that are on the market right now. The first one is extrusion printing. And it can kind of be thought of as frosting a cake or something like that, where you have a nozzle that's extruding material one layer at a time, and you come up with a final part. The other type is called stereolithography 3D printing, where you have a giant bath of liquid photoresist, which is a light-sensitive material, and you selectively expose parts of this bath to a laser, layer by layer. And once the photoresist is exposed to this light, it solidifies and it hardens. So then more resist is put on top of that, and you do the same process again where you selectively expose. And through this process, this layer by layer approach, you end up coming up with a part which can look exactly like an extrusion printed part. It's just a different method of doing it. So direct laser writing is it's related to stereolithography. It relies on focusing a laser beam into a light-sensitive material, and instead of everywhere that's exposed to the laser getting polymerized, you only have polymerization at the focal spot of the laser. So you're concentrating down all of this energy into a tiny focal spot, and this focal spot is known as a voxel. And so with this high concentration of energy, you're causing the liquid resist to solidify, to go through a chemical reaction. And when you have this happen, you can move it around using a mechanical stage, and you get really nice 3D printed structures. We're using a system called a nanoscribe, which is a type of direct laser writing printer. And there's an image of it here. You can see it's an inverted Zeiss microscope, and you can see in the center region, it's kind of dark in there, but you have a microscope objective lens, which gets focused up into your droplet of photoresist, and that's where the reaction happens. And we can get resolutions down to about 200 nanometers in the XY plane and 600 nanometers in the Z plane. And if you look at the diagram of a voxel, it's kind of like an egg. It looks like an egg shaped. It's a stretched out sphere. So there's a difference in resolution between a voxel stretched in the Z direction versus in the XY plane. And here we have a little schematic kind of giving you an idea of how a structure would be printed. So everywhere this focal spot is, you get polymerization. Here are a couple examples of structures that you can make using this technique. And to give you an idea for how small we can make this happen, this is a piece of human hair, which is about 100 microns in diameter. So if you think of a piece of human hair, you can see this text that's written on it. It says nanoscribe on it. To give you an idea for how small and tiny these structures can be. So although direct laser writing offers us the ability to make these really nice, intricate 3D structures, and they could be useful for a whole different variety of applications, regardless of the applications, there are a lot of challenges that we almost always have to address when we're using this technique. A couple examples are we need to make sure that the materials we're using are compatible with direct laser writing. We have to make sure that they're going to polymerize when the laser hits them and that they're not going to burn. We also have to figure out how to characterize the structures that we make. So it's very cool that we can make these structures, but we don't know anything about them. How do we know how strong they are, or if they're toxic to cells, or if they're going to collapse in water? So there's a lot of different properties that we need to try to figure out without using conventional techniques that you would use to measure these materials. Additionally, although 3D printing is really great for prototyping, if you come up with an idea and you realize it works very well, it's hard to scale up. So it's not ideal for mass production or manufacturing over large length scales. So if we find that we can make a 3D structure and it really matters to heart cells or something like that, then we have to come up with a good way of patterning that structure across multiple length scales. So that's something that will be challenging. Additionally, it's really hard to handle these structures. So you make them, and you might lose them. They might just get lost in the fabrication process, or you might drop them or squish them with your finger. So you have to be very careful when you're handling these little tiny plastic structures. So how does it all tie into the CellNet program? So here we have the 3-plane diagram. And there's a couple different areas where we can fit in. The first one is making scaffold materials in the testbed development. So since we're starting from scratch, we really need to be sure that we're covering all the bases in the fundamentals of the Since we're starting from scratch, we have to be sure that we understand the fundamentals of how cells behave with their microenvironments. And so even at length scales where cells are sensing things that are around the same size as they are, we need to be sure that we understand how they interact with those different structures. And direct laser writing offers us the capability of making structures like this that we can grow cells on and figure these things out. It's also important for developing new materials that might be interesting later on down the line, understanding the different nanomechanics, the properties that we can get from these different structures, and finally moving them. How do we interact with microstructures, and how do we incorporate them into larger designs? So here's a schematic of a human heart at a bunch of different length scales. You can see the little scale bars in the bottom left of each section of the figure here. So you have the top left corner, we have a human heart. It's very fibrous and muscly, and it's on the order of centimeters. And then you go a little bit further into it and you can get millimeters, and then finally, when you go all the way down, you can get a nucleus of a cardiomyocyte, which is the center of the cell. That could be about 10 to 20 microns. Just a quick reminder again, a width of a human hair is about 100 microns. So you can imagine there's a lot going on, a bunch of different length scales inside this biological system. And so we really want to understand how the cellular environment and all of these different length scales impacts tissue function, because this is what we're going to need to know if we're going to develop a cardiac patch. In terms of new materials, what sort of things are we interested in developing? We want materials that are compatible with direct laser writing, DLW, our process. It'd be great to have a high resolution of these materials. They need to be compatible with heart cells, meaning that they're non-toxic, they're the appropriate stiffness. They can't be too hard or too pointy. There needs to be a range of different mechanical and chemical properties. For example, developing sticky or non-sticky surfaces for cells. And also we have to make sure that it's self-supporting. So it's great to have soft materials because your body is very soft, but you don't want it to be so soft that it doesn't have any sort of mechanical rigidity where it can't support itself, it can't hold itself up. So these are all things that we're just starting to tap into and explore. They'll be very important later on down the line for the CellNet program. And one of the biggest challenges I think that we're going to be addressing in the next few years is how are we going to move these structures? We can't even see these structures in terms of characterizing them, understanding the nanomechanics and things like that. And in our lab, we've developed several different techniques with interacting with these structures, in addition to our collaborators at FIU and in different labs at BU and everything like that. So one example of how we can interact with these structures is through using microelectromechanical systems, or MEMS devices. And there's a whole bunch of different kinds of MEMS actuators. Actuators is just a word used. It means ways of moving things. So we also have magnetic actuators, which are more suitable for use in water. The MEMS actuators are great because they're very precise and they have nice resolution, but they're electrical devices. You can't just dunk them in water. Because we're working with cell systems, we need to be sure that what we're using would be good for use in aqueous solution. And so magnetic actuators are one thing that we're exploring a little bit more. Additionally, we have microfluidic actuators. Here is a quick movie showing you how just by changing the pressure in these microchannels, we can get a PDMS membrane to move. And so by printing structures in these microchannels, we can deform the structures and better study their mechanical properties. Finally, our collaborators at FIU have a nano indenter, which can be stuck inside a really powerful microscope, and we can watch how our structures are deforming under pressure. And so this will be very helpful in characterizing mechanical properties, seeing how it's different from the bulk material or the material at the macroscopic scale that is typically tested when you do any sort of characterization. Overall, we expect that direct laser writing will be a really nice prototyping tool that can help us better understand all of the cues that are important for controlling how heart tissue behaves, whether it's physical, mechanical, or chemical.
Hi, so right now we're at the Photonics Center at Boston University, and this is Alice White's lab. We're in a yellow room because we can't expose our photoresist to any ambient light, otherwise it will cure. The chemical reaction will happen and it will make it hard. So I'm going to show you how to use the nanoscribe, which is our direct laser write printer. To start off, we have a sample holder, which is just a piece of aluminum, and on it I have a piece of double-sided tape with some silicon. And on the silicon there's a droplet of photoresist, which is the liquid light-sensitive material that we're going to be making our structure out of. So here's our sample holder. Right here we have a piece of double-sided tape with a tiny piece of silicon on top of that, and then there's a droplet of photoresist, which you can kind of see. It looks like honey. It's a very viscous, light-sensitive material, and so that's what we're going to be making our structures out of. Piezo stage, which is a really high-precision stage that can move your sample around. We also have controllers for the laser, and then the whole laser system is in the back section here. Everything is mounted on this air table because we're on the 6th floor of the Photonics Center, surrounded by a train and a subway. So this prevents any sort of vibration while we're doing our printing, because the smallest vibrations will cause waves in our structures, which we don't want. So the next step is I'm going to turn this upside down because it's an inverted microscope, and the objective is going to dip into the material here. And now I'm going to insert the sample into the sample holder. Okay, so now once the sample's in, we're ready to print. The first step is to approach a sample and dip it into the photoresist, which is what we're doing right now. We're dipped into the sample right here. You can see the surface of the silicon. And so if I move around, you can see small pieces of dirt that I didn't do a very good job cleaning off here. So I'll load a job, which is the job file, this DNA job that we have here. And before it starts printing, the laser calibrates. So this is to make sure that the power that we're using every single time is the same. Once we have our laser calibrated, we can start the job, and you can see a live view image of what's printing as it steps up through the Z height using the piezo stage. And the whole process is extremely quick. So for this 300 micron tall structure with resolution around half a micron, we can print this in about 30 seconds. So here's our final structure. And once we're happy with it, once we do some power tests to make sure that we're using the correct laser power and we're not burning the material as we're printing it, or if there's not enough power, then we just won't get any polymerization at all and we just won't get the structure. So once we optimize all those parameters, we're done printing and we can take it out and stick it in something called a developer. So the developer, what it does is it just rinses away all the material that hasn't been polymerized. So all that liquid resist honey material is going to be gone afterwards, and we're just going to be left with the final structure. Because it's so small, you can't see it. It's on there, but you can't see it. And then we stick it in our developer solution for about 10 to 15 minutes for simple structures. You would want to develop it for a little bit longer for more intricate structures. And then when we take it out, we'll be ready to go.